Thank you, Jonathan. I'm not talking about LLMs today, but thank you for that introduction, and thank you for the local organizing team who've done such an awesome job of pulling this meeting together. I mean, it's really a pleasure and an honor to be here. My first auditory cortex was in Grantham, and I've been a groupie ever since. So I've changed my title slightly from the one in the program, because when I came back and looked at the title that I'd submitted, I was kind of reluctantly made to reach the conclusion that what we had learned from neurophysiology about auditory scene analysis was effectively nothing. So I'm going to lower expectations slightly and have a bit of realism here and talk to you about how I think, and in my lab we're sort of trying to wrestle with the question of how we might use neurophysiology to potentially actually make sense of how the brain processes auditory scenes. So I think probably everyone in this audience is familiar with this image or one like it, which was inspired from the description that Al Bregman made in his auditory scene analysis book, as auditory scene analysis being analogous to the challenge that a blind man would have trying to figure out what's on a lake if all he had available was to just dig 2 channels in the side and pop a float in each one and observe the movement of those floats. Now, Bregman's book was first published in 1990, and already within it there's a huge amount of information about how from a behavioral point of view, we separate competing sounds and perform auditory scene analysis. And I think we've made great inroads in that since then, in understanding, what are the acoustic features that allow you to pull sounds apart. But I think we've made much less progress in really understanding how neural circuits kind of achieve that feat. And much as I like this lake analogy, I think it doesn't really reflect the sort of auditory world that we find ourselves living within. So we're surrounded by sounds. And when you kind of perceive a soundscape, you don't just identify the sources that are making those sounds in the environment. You can say where they come from, both with respect to yourself, the listener, but also with respect to each other. And this is not a trivial process, right? So you have a frequency representation at the cochlea, and all of the sounds in your environment usually overlap on the cochlea in terms of their space and also in time. And somehow the brain has to kind of pull all those frequency components apart and regroup the ones that originated from the same sound source and keep them perceptually distinct from sound elements that came from another source. And that's really like the nub of the issue that we've been trying to get to in the lab. So we can kind of, behaviorally, auditory scene analysis has sort of been grouped into what we can call simultaneous cues. So if you have this kind of big sound mixture from this busy acoustic scene here, so these sort of simultaneous cues are things like common onset timing, common pitch, common location. And that's going to help you to do things like tell that some sound elements are coming from the man's voice and others are coming from the notes of the guitar. There are also what we call sequential cues that allow you to link together sounds that are actually coming from the same source. So it could be the click of these knitting needles, it could be footsteps. And that really requires that you integrate sounds over time in order, and actually potentially have a quite long timescales, right, in order to actually link these things together into what we call a perceptual stream. So integration over time is a really essential element of auditory scene analysis, and in some ways is kind of really special about the auditory system. And yet, as neurophysiologists, to make a massive sweeping generalization that obviously doesn't apply to everyone in all experiments at all times, most of what we do is to fix an animal in place and present a short sound burst and take a snapshot of the neural activity that occurs after that and try and deduce something about that short sound. And these sounds are often separated by quite long periods of silence. There is no scene analysis problem here, right? You have one sound and a Coke chamber, whatever. So we've been trying to move from this kind of snapshot approach to understanding scenes. So our approach, broadly speaking, rests on having good behavioral tasks, thanks to AI, at about 10 p.m. last night. Kerry saw all of the iterations of this image. The glass of water was particularly challenging for it. So we train animals in tasks that we hope kind of pull apart elements of auditory signal analysis. We maybe can't do like the whole poster room downstairs with our ferrets, but we try and take elements of the real world and bring them into behavioral tasks. We're rooted in auditory cortex, but also interested in, I guess, what you might call extra auditory cortical areas. So areas that receive information about sound, such as the hippocampus or frontal cortex. And if you missed Jair's poster yesterday, this is what he looks like. He was presenting some beautiful viral anatomy data. You should track him down at coffee. And as Jonathan pointed out, all of our work is, or all of our animal work at least, is using the ferret as an animal model. I feel like most of you know why we use ferrets, but for us, the low-frequency hearing is really critical. I mean, Patrick pointed out their beautiful aroma and their early development, but for us, it's really the fact that they perceive interaural timing differences in order to carry sound localization cues. They're capable of pitch perception, and these are the kind of low-frequency phenomena that human listeners use to separate competing sounds. They're also relatively large, so they have a number of auditory cortical fields, and they're big enough that we can target each one individually and know which one we're in. And they're also really good for behavior, so they're quite smart. Maybe not as smart as we like sometimes, but smarter than a mouse, I would argue. So what I'm going to do today is tell you some short stories of different experiments that we're doing in the lab at the moment that are all, almost all unpublished, almost all in not kind of final complete states, but they should give you a flavor of what we're doing, and hopefully, in 3 years' time at the next auditory cortex, we might be able to say something about how the brain's actually doing these things. So I'm going to start off with a story about how auditory cortex separates competing sounds, so target sounds in noise. And we're going to use a simple auditory scene. So having told you that it's, silly to present sounds in isolation, that's more or less what we're going to do. So we're going to train ferrets in this task to discriminate the identity of artificial vowel sounds. So on each trial, they hear 2 repetitions of an identical vowel, which could, for example, be oo, oo, or air. And they hold their head at the central spout, they'll hear those sounds, and then they make a left-right judgment according to the identity. So they might go left for oo and right for air. We can embed those sounds in background noise, and the sort of background noise that we're using is simple broadband white noise, but it's got a temporal time course that comes on with the onset of the first vowel and gates off with the offset of the second vowel. And I think the time course of the noise would go quite a long way to explaining some of the differences that I'm going to show you from the work that Jean-Marc was presenting yesterday. It's data that was collected by Stephen Town and has been more recently analysed by Jules Lebert as part of his PhD thesis. So we've done a lot of work with these vowels, starting actually, when I was a postdoc in Oxford working with Kerry and Jan and Andy. We like these artificial vowels because they're broadband, so they activate a large number of neurons in auditory cortex, and we can kind of parametrically control different features of them, like their identity by shifting around the formant frequencies, their pitch by changing the fundamental frequency, and things like, obviously, their position in space or their level. And we've done a number of studies that mean that we've got a fair idea of how they're represented in auditory cortex and which kind of cortical fields we should be looking in to find timbre-sensitive responses, which tends to be the kind of tonotopic primary and secondary areas. And we've learned over a number of years that behavior itself dynamically modulates the way that information is processed in auditory cortex. So like many other labs, we see that when we engage an animal in a task, the firing rates that we observe are suppressed relative to a passive state, but that the information content itself is maintained or even enhanced, which is what you can see on the bottom here. But one of the kind of, I think, really interesting hallmarks of behavior that we saw was that animals engaged in discriminating timbre sounds actually accelerated the information content of their responses compared to when they were passively listening. So it seems like when you engage in a task, the informative kind of elements of the neural response get kind of pushed further forward in time, and that's not true for the task-irrelevant features. So for example, like if the animals are discriminating the identity of vowels that vary in their pitch, we can record sensitivity to pitch as well as timbre, And that time course is not changed by the animal having to kind of ignore pitch. So we have these hints from previous studies that we've done that there are kind of dynamics in auditory cortex that can be modulated by different tasks and that could be kind of interesting to dig into. So we trained a reasonably large cohort of animals in this vowel in noise task, and you can just see here plotted the effect of varying the target sound either in a background of silence or a background of noise in orange, and unsurprisingly, noise impairs performance in a signal-to-noise dependent manner. No real surprises there. And silencing auditory cortex in our hands, in our animal model, in our task, has a clear effect on behavior, whether you do that with optogenetics or with cooling. If we silence in silent conditions, when there's no background noise, then auditory cortex is reasonably dispensable. We don't see any impairment in that case. So we went on and recorded neural activity from the kind of low frequency area where primary and secondary cortex meet in the ferret, and we observed all sorts of neural responses. There's just one shown here. So this is a neuron that clearly differentiates the identity of the vowel based on its firing rate, and it seems to do so in the presence of background noise more or less as well as it does in silence. This neuron is probably one of the exceptions. So this is a kind of population overview of all of the neurons that we recorded just in terms of their firing rates in the clean condition and in the noisy condition. And I think the sort of most striking difference here that you can see is this kind of robust onset response that is presumably driven by the noise. And if we take a kind of differential view to say, to look at the difference in activity between the two vowels, which is what the animal's discriminating, we can see in clean conditions we have neurons that respond better to the oo at the top, so that would be a neuron like this, and the blue kind of neurons are preferring the other vowel. And that representation, for want of a better word, gets noisier. In the noise condition, you can see less kind of structure here. Single unit decoding kind of supports that, so there's a small fraction of cells that can maintain their decoding performance in noise, although the overall level of decoding is lower. And there are plenty of neurons that can encode vowel identity well in the quiet condition, but not when you have background noise. If we take a population coding approach, so here we have kind of 31 simultaneous populations of between about 15 and 20 neurons, then the representation in the face of noise becomes more robust. So now we have a majority of these populations that can encode the identity of the vowel in the presence of noise, but still, like the behavior, importantly, there's still a kind of hit in performance here. So there's two ways that you can kind of, I guess, view this sort of data. And the first is to take a sort of signal processing perspective, which is that we know that noise is going to corrupt the peripheral representation. And there's also kind of evidence that auditory cortex might denoise representations through things like contrast gain adaptation. Alternatively, and by no means mutually exclusively, you could take a kind of auditory scene analysis perspective. So here, noise is just another element of the auditory scene. And if you can segregate the scene into its constituent components, then you're going to be able to focus on any one sound more effectively. So with that kind of in mind, we turned to trying to understand more about how the dynamics of the activity in auditory cortex evolve over time in this task. So what we're going to do now is consider the activity of all of the neurons that we recorded, which we have done dimensionality reduction on, and I'm just going to show you the trajectory that the activity takes through those first three principal components. So this is the activity before the vowel comes on. This is the onset, sustained, offset response, silent period between the two, the onset of the second vowel. I'll let it play around again. So there's a clear kind of trajectory that activity is taking here in which the identity of the two vowels is separable. And you can see the structure that each of the two repetitions of the identical sound kind of creates. So the question then is what happens when we add noise into this? So now the noise is plotted in orange. And the first really main thing to note is that at the onset of the noise, you get this huge kind of deviation into a kind of different part of the population space. So this is projecting the noise data into the dimensions of the clean data. But after that onset, the activity kind of converges back to something that looks really quite similar to the trajectory that was being taken by the clean data or by the vowels when they're presented in silence. So in order to quantify this, we've used cosine distance metrics. So this is essentially asking in this high dimensional space, what's the difference in the kind of angle that the population is taking at any one point in time. We can measure the cosine distance between any single time point and every other time point, and that can allow us to kind of pull out neural states where activity is broadly similar within a state. So what we can see here is the pre-stimulus activity, which is similar to itself, and not that similar to any of the other kind of elements of the task. So there are 3 silent intervals here. There's the pre-trial, the between the 2 vowels, and the after the second vowel when the animal's registering its response. And this pre-stimulus activity, you can see it as a distinct epoch of activity in this space. We can go on and look at the onset of the first vowel, and you can see again that the onset of the vowel has its own distinct state that is actually different from the sustained activity. And the onset is very similar to the onset that you see to the second vowel. And again, we can keep kind of tracking through the elements of the task here, and what we're left with is this quite clear checkerboard that shows us that each of these kind of time windows of the task has its own state associated with it. And there are some periods that are similar, like the vowel onsets, but largely you get quite a kind of clean representation. When we do the same thing for the noise trajectories, you can see that all of these states are less well defined now. So all of the distances are a bit closer in this space. It's as if the neural activity is sort of somewhat collapsed in. But you can also make out this kind of clear square, which is the time course of the noise itself. So you could see this as potentially a representation of the vowel and the noise in this space. We can compare directly these 2 metrics and end up with something that looks like this. So now we're measuring the distance between the clean distances and the noise distances, and they kind of peak in this intervowel interval when essentially there is noise in one case and not in the other. So that difference is presumably the representation of the noise. We can do the same thing with Euclidean distances. So that tells us not necessarily whether the population is going in the same direction, but if it's traveling the same distance. And here you see that there's a huge, as you saw in those trajectories when I showed you to them, at the onset, there's a really big departure that happens as the whole population is kind of responding to the onset of the noise. So we essentially wanted to try and come up with a way of quantifying how much noise was contaminating the response, with the kind of hypothesis that if auditory cortex is treating this as a scene analysis problem, the common onset is going to drive activity that is kind of fusing the sounds together, and then over time, if you can orthogonalize the response to the 2 sounds, you can effectively pull them apart, and the animal could then, if it were patient enough to wait to the repetition of the second sound, make a better decision on that. So the metric that Jules came up with was to essentially, so now we're stepping back from using all of the neural activity, and instead we're going to use the simultaneous populations again. And for each population, we're going to do dimensionality reduction on the clean data, and we're then going to use linear discriminant analysis to define a decoding axes in this space that best separates the 2 identities of the OO from the air effectively. And then we're going to project the noise data into that space with the hypothesis that if the noise data is orthogonal to or if the effect of the noise is orthogonal to the vowels, then it shouldn't change the distribution of data on that decoding axis. Whereas if there is contamination, we might expect to see the population activity kind of shunted in one direction or another. So we can kind of visualize that for just 2 points in time here. So at the onset of the vowel, we see exactly what I've just described. So by definition, the middle of or the mean of the activity in the clean condition has to lie on the decoding axis, at least if the decoder's any good. But in the noise case, you can clearly see it's shunted away from it. So here we would say noise is contaminating this response. Whereas by the end of the sustained activity, the noise data is slightly closer to the decoding axis, but it's still symmetrically distributed around it, suggesting potentially that the noise has kind of been orthogonalized in this case. We can take that sliding window approach in 50 millisecond chunks throughout the course of the trial, and we get something that looks like this. So it seems that indeed onsets drive things to be together, and you get very high contamination metrics. And interestingly, the same thing happens when you get the onset of the second vowel. But after the onsets, you get this gradual decay where the noise is effectively kind of separated out from the target sounds. Okay, so kind of interim summary here. Auditory cortex is necessary for listening in noise, at least if the noise is gated on and off with the target sound. So we do have other data which shows that if noise is continuously present, then the effect of auditory cortex silencing is much smaller. And we see that auditory cortex might adapt to background noise by effectively orthogonalizing the response to noise. There are some limitations here, though. The target sound is still brief, it's still transient, it's still quite simple. There's very little here to actually promote stream formation. And the noise itself is quite simple. So our sort of response to this, which has been a project that's been brewing, I think, for nearly a decade, slightly embarrassingly, is a task where we've really kind of upped the complexity. So now instead of asking animals to discriminate artificial vowels, we're going to ask them to discriminate human speech. It's not because we think they understand human speech, but it's a nice spectrotemporally complex sound that we have good access to. So now the animal's trained to listen for the word instruments, and the word instruments is embedded in a stream of other words from a library of about 100. It can occur anytime between 1 and 6 seconds in this stream, except on catch trials when the animal's rewarded for waiting at the central spout. And if the word instruments occurs, then the animal has to move to the side from which the whole of this speech stream is presented. And this is a behavioral task that Joe Cellini, who's here somewhere, helped set up when he was in the lab, and Carla Griffiths and Jules Lebert have really been involved in establishing the behavior for over the last few years. So I'm going to play you a video of what that looks like.
Any about the tools and the mice and the small instruments.
She's getting her water reward.
The generations faces the tools, but the stems instruments did.
So there's no grammatical structure to this, right? They're just random words drawn from a passage on shipping, which is why there's a story, but I don't have time for it. And if you got to the animal in the cage and go, instruments, they look at you with a kind of, what expression? So anyway, animals can do this task. So they do it quite well. If you look at the overall percentage correct, it's quite high. Their hit rates are high. Their false alarm rates are low. Their corresponding D primes are robust. In this case, we've got data plotted here in blue for the natural. So they're trained on one male and one female talker. Data I'm not showing you is that they will actually generalize to other unheard talkers. But in this experiment, we were looking at how pitch is used to actually link sounds together into streams. So we had two manipulations in this task. One we call the intertrial roving, and here the whole stream is shifted up or down in pitch. And we have what we call intra-trial roving, and here we rove the pitch of every individual word. So this should kind of hinder stream formation, right? And as you can see with these behavioral data, actually the animals were quite robust to most of those perturbations. But underneath this kind of high-level overview, boiling down tens of thousands of trials into single D primes, there's a lot of complexity. So we've kind of been toying with machine learning models to really try and exploit the power of these massive data sets that you get when you highly overtrain your animals on tasks for years and years and years. So in this particular task, the sound can come from the left or the right. The talker can be male or female. We can have noise or not. As I say, the F0, the pitch can be natural or roved within the trial. And all of those things you might hazard would affect performance, and those of you who work with animals know that other things will affect performance, like the time of the target word in the trial, the stimulus history on the previous trial. So we've been using gradient-boosted models, so gradient-boosted regression for continuous variables like reaction time, or gradient-boosted classification for things like hit rates and false alarm rates, in order to capture the kind of complexity of these behavioral data sets. And the advantage that they offer over things like traditional generalized linear models is that they allow us to really look at both nonlinear variables, but also non-monotonic ones, and also the interactions between them, which we know are often really important. And the output of such models are things like this, so things that tell you I guess this would be analogous to a beta coefficient in a regression model. It tells you how large the influence is. We can run kind of permutation tests to check that things are significant. But then you can project things into plots that look like this. So this is a Schaap's value plot. So here we plot every single data point in the data set, weighted by the contribution that that particular feature makes to the reaction time on that trial. So here we've got data on correct trials only, and we're looking at the reaction time that the animal makes to the word instruments. And we can see that things that we would expect affect the model, like the identity of the ferret, also the identity of the talker, which is quite trivial because the female voice is slightly shorter than the male. So the reaction time is faster, but it's a good sanity check. We can look at whether pitch roving has an effect, and basically it doesn't, except for the very lowest pitch of the male voice, which I think butts up against the lowest frequency edge of the ferret's hearing. And interestingly, we can pull apart here a really nice clear effect of pitch roving on the animal's reaction times. So when the pitch is consistent within a trial, the animal's reaction time is faster than when the pitch is roved. So this is really nice evidence for us that the animals are streaming sounds together in a way that's very similar to how we as human listeners would do that. We've got quite a large cohort of animals now trained and implanted with recording electrodes in frontal cortex and auditory cortex. And I would direct you to Jeffrey's poster tomorrow, because he's going to be, or Thursday, he's going to be presenting that work. We're generally quite interested in streaming and competing sounds and even visual influences on them. And I'd also direct you to Rebecca's poster on Thursday if that's your cup of tea. So I was going to tell you about a story now about kind of how, looking again now at a different timescale, how statistical regularities are extracted by auditory cortex, but having looked at my talk yesterday, I kind of decided that you didn't want to sit here for 3 hours. So I'm going to give you a brief commercial break, because Kat, who did this work, Kat Poole, when she was a PhD student in my lab, is in the audience. So if you're kind of excited by it, you can track her down afterwards. But this was work that we did in collaboration with Maria, using Maria's stimuli, if you're familiar with them, where essentially you have repeating tone sequences that elicit this perception of regularity, and we trained animals to detect this regularity. We've been recording from auditory cortex and trying to tease apart things like signatures to regularity and stimulus-specific adaptation, but that's a story for another day. So with the last little chunk of this talk, I'm now going to turn to the kind of spatial aspect of auditory scenes. So we know that localizing sounds is critical for survival, it's important for listening and noise, all the things we normally say. But we've become really interested in coordinate frames. So to kind of unpack what I mean by that, if you're this listener here and you're listening to, I don't know, a conversation over here that this person's having on their phone and the sound of the coffee machine here, you can describe where those sounds are in one of two reference frames, or one of at least two reference frames. So the first is what we tend to think of as auditory scientists, so a head-centered reference frame. So here the person talking is behind you to your right, and the coffee machine is to your left. But you can also describe the position of the sounds in a world-centered reference frame. So here the person who's talking is by the window, and the coffee machine is on the table near the wall. And we first got interested in this, and this was work that Stephen Town did a number of years ago, because Stephen developed some simulations where he modeled effectively a head-centered unit that had a classic kind of contralateral or to the right of the head tuning, and a world-centered unit that, if you imagine these red things are speakers, likes sounds that originate from the east of the arena. And if you generate simulated spatial receptive fields from then an experiment such as most of us do them, where you fix an animal's head in the middle of a speaker ring, then you end up with spatial receptive fields that are indistinguishable from one another. So effectively you have a coordinate frame ambiguity here. The way to break that ambiguity is to allow the ferret or the listener to take a variety of different head positions. And then what happens is that as the head, as the head rotates through each 90-degree increment, then the tuning curve for a head-centered unit, when plotted as a function of the speaker position in the world, systematically shifts, whereas the world-centered unit remains a single overlying spatial receptive field. And the same thing is true, or the inverse is true if you plot firing rates as a function of angle relative to the head. So here the head-centered unit has a single spatial receptor field, and the world-centered unit has a systematically shifted one. So back in the pre-deep lab cut era, we mounted little LEDs on our animals' heads and tracked their position as they navigated freely in a ring of speakers that were presenting sequences of clicks, which allowed us then on each and every point in time to work out the angle of the animal's head with respect to the speaker. We, of course, know which speaker presented the click, so we can then map spatial receptor fields in both of these reference frames. On the left is the simulated responses, just to kind of remind you what they look like, and on the right are just 2 example neurons that we found that fulfill the kind of predictions of those simulations. So a kind of head-centered unit that made up about 80% of the spatially tuned neurons that we recorded, and a world-centered unit that made up about 20% of the neurons, spatially tuned neurons that we recorded. So this work is a bit old now, and we've built on it in a number of ways. So firstly, we trained animals in 2 different tasks. One was a world-centered task, and one was a head-centered task. And we were able to show that animals could discriminate sounds in both of those reference frames. I'm going to tell you about the head-centered task now because we have some neural data that I'm going to present. So in the head-centered task, the animal stands on this central platform, and it's trained that when a sound comes from in front, it should respond to the right, and when a sound comes from behind, it should respond to the left. The trick, such as there is one, is that between testing sessions, the position of the central start platform is rotated, and we keep the target speakers in alignment with that rotation. So the sound is always doing front, right, back, left. And when we record neural responses, we see we see many neurons that look like this. So here's a neuron that fires in response to the sound that comes from in front of the animal, irrespective of the direction in which the animal is facing. Here's a kind of counterexample where the neuron fires to the sound that comes from the back when the animal's facing in this direction, and it fires to the speaker in front of the animal when it's facing in this direction. So this is consistent with a world-centered representation, so the neuron is firing to sounds that come from the north of the arena. Now, in addition to the target sounds, which come from the front and the back, we have these rarely presented probe sounds from all of the other locations. We can again turn to simulations to make predictions about what each of these 2 models would generate in terms of the kind of cortical representations. So here, if you imagine a spatial receptor field for when the animal's facing forward, you get a kind of slice through this 2-dimensional world angle and head angle space. And of course, because the animal is only in one location, the position of the two reference frames is completely covariant. If the animal rotates through 180 degrees, we get another slice. And if we allow the animal to take all 12 30-degree increment positions, then we get this 2D representation. We can do the same for the world-centered unit, and really what I'd like you to appreciate here is just that the head-centered units are horizontal and the world-centered units are vertical in this space. So with that in mind, here are 2 example auditory cortical neurons, one of kind of each flavor. So we have, again, many units that show head-centered firing, but a minority that show these quite clear world-centered tuning. This is data analysis that's really ongoing, but kind of our preliminary kind of snapshot of what we've got so far is that the proportion of world-centered units we see is very similar to that we observed in passively listening animals. And I mean, for us also just replicating the fact that we see these world-centered representations in an active behaving animal is really reassuring. So I'm gonna, in the kind of dying minutes of this talk, turn to kind of one final aspect of sound localization and real-world listening that we haven't yet touched upon. And I'm going to illustrate that with this, because who doesn't like a bit of David Attenborough?
60 million tons of
Sorry,
snow
could we turn it
now
down a
blanket.
bit? Thank you. And I need to get to the right point, because much as I feel like we might enjoy an hour of David Attenborough, we probably don't have time for it. Okay, so he's hunting a vole under the snow. It's very exciting. he's got breakfast.
A vole.
So what I hope you appreciated, if I didn't manage to not start the video in quite the right point, was that when the fox was planning its kind of motor trajectory to catch this vole, it made a really characteristic series of head movements, right? It was scanning, presumably improving its binaural cues by making these very active head movements. And that's an aspect, I think, of real-world localization that we really try really hard to eliminate often in the lab, right? We really train our animals, even our humans, we give them a chin rest. And yet, I'm sure it seems like it's an essential component of active localization. So we have recently developed a setup that allows us to kind of get closer to looking at this. So in this experiment, we have a large arena, so it's 4 meters by 2 meters. You're viewing it through a fisheye lens, which is why it's kind of wonky. And what you're looking at is an acoustically transparent floor. And under that acoustically transparent floor, we have 48 speakers. So we can generate all sorts of acoustic scenes on this grid. And at the moment, we're generating a very simple scene where essentially the animal is trained to go to this start location here. That's going to trigger a sequence of noise bursts whose timing you'll be able to visualize by a little LED flashing here. There aren't any sounds, unfortunately. And the animal is trained to navigate to this location. When it gets here and traverses this location, the position of the sound is going to move. So it's a kind of 2-stage task where it has to track a sound across this grid. And when it reaches the final location, she has to pause there in order to register her response, and then she'll get a reward. So there she goes, the sound moves, she comes back to it, she waits, she looks up at the ferret god in the sky, and she goes off and gets her reward. Another example, different ferret, slightly more ponderous. So the task is controlled by Deep Lab Cut Live, which is why you can see these markers on her. She goes there and kind of patiently waits, and then the reward activates, and she goes off and gets her reward. So together with collaborators in Bonn from Jason Kerr's group, we've fitted this chamber with 8 high-speed recording cameras that allow us to get multiple 200 frames per second views of the ferret. And when we're doing these head tracking experiments, the ferret wears a kind of little infrared set of antlers that mean that we can localize her head position in 3D. And from that 3D representation, we can begin to look at how the head is actually being moved to actively sense the sounds in the different components of this response. So here is just some kind of preliminary data that we've got from one session. So the black line that's marked here is the point where she traverses the first sound location and the position switches. So effectively what you're seeing is her zooming in, honing in, this is a kind of shorter time scale, honing in on the target sound and then having to reorient and figure out where the second sound is. And I think there's some really interesting stuff here that we're beginning to unpick with the animal kind of making sort of horizontal head movements as she approaches the target to kind of zoom in on it. At the target, there seems to be a change in elevation, which possibly relates to the fact that the speakers are under the floor, rather than the fact that she's looking up at ferret gods, really. And yeah, like some preliminary data show that as the animal, so this is basically looking at the sorts of the change in head angle that we see when the animal's a different distance from a target. So here, as the animal gets closer, the yaw, which is the horizontal head movements, increases as she gets towards the target, whereas the other two are quite stable. We have a poster on this on Thursday that I'll give you the reference for in a minute. So in some experiments, we do head tracking. In other experiments, we record with bilateral neuropixels that are implanted in auditory cortex in a way that targets auditory cortex and potentially the underlying hippocampus. So we can also record neural activity while the animals are doing this task, and we can end up with sort of plots that show us for any point in time, at any position on the grid, when a sound was on, what was the firing rate, and obviously by then calculating the animal's head angle, we can begin to construct kind of classic spatial receptive fields, PSCHs, and things that look like the sort of snapshot approach that we're used to trying to make sense of. And if this is something that you're interested in, then I direct you. So Stefan has a poster this afternoon on the neural recordings, and Amit has a poster on the head movements on Thursday. It's all quite preliminary, so we'd love to hear feedback on it. And that is actually my last slide, because my conclusion is that there's a lot of work to be done. And I feel, we've spent quite a few years actually developing these behavioral paradigms, and we're only just beginning to get stuck into the neural responses. But I hope that we've kind of given ourselves a good shot of actually coming back in 3 years' time and having something meaningful to say about how neurophysiology has contributed to auditory scene analysis. So the people who contributed are all shown here, as are our funders. So thank you to them and to you guys for listening.
Open for discussion. Questions?
Oh, I'm so proud of myself. I caught it. Is this working? Thanks. Yeah, that was a nice sort of whirlwind tour of so much great stuff. I guess a lot of questions, but the one thing that I was curious about with the first test where you're doing the vowels and noise, one of the questions we've run into is kind of understanding. You can use a decoder to say sort of optimally how can you get information out of the brain, but you always sort of wonder, is the animal actually using it? And I guess I was wondering if in any of the analyses that you've done, if you've seen any evidence about how you can relate does the decoder that actually discriminates vowels successfully, can you relate that to the signals that the animals are actually using in the behavior?
Yeah, I mean, it's all correlational, right? We can look at things like whether we can decode. So, some decoders you can get choice activity out, which gives you a slightly closer coupling to the neural activity in the animal's discrimination, but fundamentally I guess we have kind of two strands, which is does the brain area contribute if we silence it, and what sort of neural correlates do we see there? And it's beyond kind of the correlation. It's really hard to know whether the way that you're decoding it is in any way biologically meaningful, right? I think that's a problem we as a field should wrestle with.
Right, because this idea of orthogonalization, it seems like there might be a specific population that's robust and actually correlates more. But I guess so is there an alignment with the choice probability and the optimal decoder that you can recognize?
Is it? So there is at the single neuron level. We haven't yet done it for the population, and that's a good question. But we'd have to, there's sort of constraints on what I think is sensible to do with all the neurons we've recorded that come from a bunch of different sessions and animals versus the single session stuff, but we could certainly do that for the single session stuff, and we should.
That was really nice, thanks. I've been following Stephen's arena stuff, and I've just now realized that it reminds me of the old J.N. Sparks papers, where they have monkeys fixating at different locations in auditory receptive field shift. So I'm wondering about your mixed receptive fields in that. Are they, what are their nature? Do you think they're related to that in some way? And do you think they're somewhat special in terms of localization and sort of arbitrating between head and world-centered? Speculate?
Yeah, I think that's a really, really interesting question. So some of our mixed representation, the reason that I didn't make so in the first paper we saw very little mixed representations. My feeling, having seen the data, is that some of the mixed representations are just poorly spatially tuned, and they're not so interesting. We have got some hints from across various experiments that there are kind of head direction in the world signals, which could be how you get these kind of mixed responses, as if you've got a head-centered tuning curve that's modulated by head direction, then that's halfway to being world-centered. So it's something that we want to look at. In terms of eye-centered receptor fields, which is also a really interesting question, the next level of these kind of head movement studies on the speaker grid is to add eye cameras and actually measure eye movements, because I don't know that, certainly ferrets chasing visual stimuli optimize their eye movements to stabilize the sort of minimal optic flow path, rather than necessarily make directed movements like a primate would to track the visual target. So my intuition is that they're not really making massive eye movements to the sounds, but we need to know that, because actually, if there are eye-centered representations in there as well, and there may be the mixed ones, then, it's really interesting. way of looking at it.
Are there any more questions?
Triumph. Am I on? I'm loud anyway.
Hi, Fred.
Hi. Did you look, so when you had the Ferrets Learn instrument, were you able to look at changes over time in terms of the neural representation, for lack of a better word?
That would be super interesting, but I think with our neuropixels now, we can record for a year, and we've got the training down to less than a year, which is what it took initially. So in theory, it's now an experiment that could be done, but we have not done it, just because it's just a lot of risk, right? To kind of implant an animal before you know if it's going to learn the task when the task is that hard. So
I guess as a corollary to that, early on in training, did you find that the changes in like pitch and so on that were not diagnostic for instrument, did they get more distracted by those? Was it?
So that what tends to happen when you give them something new, so either new voices or new pitches, is for one session they completely throw their toys out of the pram and refuse to do anything at all. And then they come back the next day and they're fine. They've made their point. Okay. So yeah, we didn't have to train them to generalize. They did it. Once they got over that initial rage that we had changed the task rules, then they were perfectly capable of making the sensory discrimination.
Cool. Oh, damn.
Yeah, thanks, Jenny. I have a question. I mean, so you have raised this issue so that they are rolling their head and the other aspect the other thing they could use is pinning movement. Are you also considering? I don't know. To which parrots do this? I mean, I know cats do it. Monkeys also do it. We don't do it. I guess most of us, at least.
No, I mean ferrets do it in the same way that we do, which is like if they try really hard and make a weird facial expression, you can just about move them a little, but they don't make kind of cat-like directed pinner movements.
But I mean that's basically related, whether it's head related or whether it's like in the eye in the visual system, whether it's eye related or related to the head. So are you?
Yeah, we're not monitoring it at the moment, but I guess the way that the field is going with kind of face movements being so important, I foresee in our future somehow trying to rig up something that gives us access to them.
So
I don't think they make any movements, though. I'd be really surprised if they were making ear movements.
I see. Okay.
Hi, thanks, Jenny. I was wondering about these world-centered neurons, because presumably, like the auditory system, the ascending auditory system doesn't have a map of space, right? So do you have any evidence that those are actually getting information from other parts of the brain, like maybe hippocampus, like maybe are the latencies bigger for world-centered?
So I guess the first thing to bear in mind is that even in the cochlear nucleus, you get vestibular input. I think Ellie showed that a few years back. Which would potentially allow you to disambiguate self from source motion. In the original data set, there was a trend for the latencies to be longer in the world-centered receptor fields. We didn't have the numbers to pull that apart. I'm hoping as the kind of spike sorting of this data set unfolds, we might get a bit more statistical power to be able to look at some of those things, but we could record head and world-centered units on the same electrodes in the same or adjacent electrodes. So there was nothing that really marked them out as like this layer or this depth. But the tracing experiments we're doing now show that there's projections from hippocampus, from places that should carry head direction signals. So I think everything is potentially there. And there's data also in the rat that auditory cortex has like head direction signals and position signals. So we're not the only people seeing things like this, but how much of it is bootstrapped onto the inherent spatial system within the brain and how much of it is coming up as some kind of vestibular auditory, essentially visual integration, I think is a good question, which we may hopefully answer. I think she had that one.
I just wanted to ask you about the allocentric versus head-centric. How much of that can be trained and how much is this genetic? Because humans in certain cultures are extremely head-centric. And in others, they can be extremely autocentric. In Japan, all maps are basically relative to your head, where you're looking. And the iPhones also do that often. And in some Polynesian cultures, you know, you get out of bed with your east foot or northern foot. And they really use absolute coordinates all the time. So I just wondered if this is the same with these ferrets. If you train them somehow, I don't know how you do that. It would take more than a year, probably.
Yeah. No, so we have this cohort of animals, right, that were trained, half were trained on a head-centered task and half were trained on a world-centered task. The animals could do both tasks perfectly well, so long as you made them make the responses in the same coordinate frame that the stimuli were being discriminated in. That was important. We have a lot of neural data from the head-centered animals because they made their responses on the platform. They did many more trials. So for kind of just reasons of statistical power, we've started with the data analysis from that cohort. But we collected all of this data in COVID and didn't have the bandwidth to analyze it. So that's exactly the question we want to get at. You know, if you train, so the fact that we're seeing world-centered representations in the head-centered animals is really reassuring to us because it suggests that at least, you know, they should be the same or more in animals trained in world-centered tasks. So.
Maybe one.
If I may ask one question as well, Jenny. So it's kind of nice that you're exploring real-world situations. And I had sort of anticipated that you were going to provide us with a snowy version of your grid to see if the ferret could leap into it to catch the appropriate sound. But I mean, we do know that ferrets, for example, hunt in tunnels, like, you know, hunt for coney or rabbits. And I'm sort of intrigued by this question because, as you know, snowy owls and the foxes are getting not only azimuthal cues, but also getting cues about elevation. So have you thought a little bit more about how you could make your grid scene a little bit more real-world realistic? In other words, can you clarify a little bit more what the actual sound that the ferrets are looking for is, and whether or not you might be able to incorporate other cues such as elevation to sort of see whether or not how they're pulling this together? That might give you more information about head cocking or head movement as well.
So the sounds that we're using at the moment are either just broadband noise, or in a handful of experiments, we've filtered the noise to eliminate either low or high frequency cues. So I think they're certainly indistinguishable anecdotally in their performance if you give them kind of low pass ITD containing stimuli. They were not too happy with narrowband ILD stimuli. How we would, so and at the moment all of the sounds are under the floor, right? And what that buys us is the fact that the animal can navigate amongst them instead of having a ring. Because you know, we don't just have sounds around the edge of our environment, but it is peculiar, right? Because all of the sounds are underneath them. And I don't know if I would not be confident that there is a really good way of projecting sounds at some higher elevation without adding in speakers, which like maybe we can do in the future, but for the moment it feels like we've already bitten off slightly more than we can chew to make sense of the data.
All right, well, anyhow, thank you very much. Very nice presentation. Cheers.